Hey guys, welcome back to my channel. So today we're going to talk about enzyme, which is a super high yield biochemistry topic on MCAT. So part one is just the basics about enzymes. We're going to briefly talk about enzymes function, what are enzyme made up of, some characteristics of enzyme, and very importantly, the relation between Vmax, Km, kcat, all of these things. And then the other two videos, especially part three, is really the highlighted content regarding enzyme, which is the Michaelis-Menten kinetics. Okay, let's jump right into part one. Okay, so what does an enzyme do first off? So we know that there are a lot of chemical reactions happening in the biological system all the time, right? A lot of reactions happening in our body, but most of the time, if we're not running a fever, our body temperature is 37 Celsius, and then we also have a relatively stable pH value range in our body. So that's not a perfect condition for all sorts of reaction to happen. So the enzyme comes in play here to be a catalyst, meaning that the enzyme catalyze reactions. Okay, excuse my handwriting. So being the catalyst of the reaction means that enzyme is not really a reactant that get to be turned into a product, right? It's it's always recycled, right? All right, so here let me just draw you the graph to show like how does enzyme help with the reaction. How does it catalyze the reaction? So here's a delta G graph, and then if this is my original reaction without enzyme, with the involvement of enzyme, you can see that I still get the same delta G, same product, but the activation energy this is the activation energy barrier, right? Compared to without the enzyme, it has been much lowered, okay? So it gives the reaction a faster rate to say. Okay, all right. So if here's a multiple choice question for you. So I say, okay, so I was doing an experiment. I put this and that XYZ enzyme into my reaction bowl, and then this change the, let's say, the delta S, the entropy of my reaction. Because of the involvement of an enzyme, my entropy delta S increased, okay? Or B, if the involvement of the enzyme would change my enthalpy of the reaction. Or C, if this makes the reaction more spontaneous, aka delta G decreased, it becomes super negative, right? So would you choose A or B or C? No, right? They're all very wrong, okay? Why? Because all the A and B and C, all the factors here are the thermodynamic factors of the reaction, right? Whereas enzyme is really related to kinetics, right? It faster the reactions rate, but it doesn't change the delta G. It doesn't make it more or less spontaneous, right? The enthalpy, entropy, they're just the way they are, okay? So just want to mention that, okay? All right, and then number two, what is an enzyme made up of, right? And then usually we were taught that it's protein, right? Okay, perfect. And then MCAT, you can also see sometimes it's RNA, okay? So I just want to point that out so you don't feel surprised when you see that. Okay, and then some other characteristics of the enzyme. Once again, I want to point out that it is recycled, okay? And then number two is I want to say that enzymes are really reaction specific. Okay, so by saying reaction specific, I mean specific enzyme engage in specific reactions, okay? So let's just do a couple examples. What about kinase, right? What does this enzyme kinase do? Okay, it would usually add a phosphate group to the amino acids like theriorine and what is the other one? Serine, right? The ones with OH, alcohol groups. Okay, so kinase it belongs to a larger class, which is called transferase, because it basically transfers the phosphate to other compounds, right? So transferase is one type of enzyme. There are 6 types of enzyme that you need to know, okay? MCAT. There are transferase, hydrolase, isomerase, ligase, lysis, and then another one is called, I believe it's oxidoreductase. Okay, so these are 6 types of enzymes. You can just Google six types of enzymes on MCAT, and then you will know. Okay, and then kinase is a kind of a transferase. But what about the opposite of kinase? Phosphatase. Okay, so phosphatase removes the phosphate group, right? So it's a type of hydrolase, right? Which means that this kind of enzyme use water to cleave a molecule, breaks the covalent bond with water, okay? To remove the phosphate group in case of a phosphatase as a hydrolase, okay? So yeah, make sure it's really easy. I don't want to just make this video super long, so please just Google 6 types of enzymes, okay? All right, so now let's come to the highlighted part of this video. The Vmax, Km, and kcat, blah, 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 okay? All right, so I want to first just write out the enzyme equation. So enzyme plus substrate, they form this enzyme substrate complex, right? And then after a bunch of reactions, the enzyme substrate complex will be turned into enzyme and the product, right? Once again, you see my enzyme is always there, right? It never gets changed. No function groups are added to the enzyme. They're not losing their functional groups. They're just the same as usual, okay? The same as always. All right, so here's my enzyme. Okay, this is the enzyme, and then I call this active site, and then I can have some substrates looks like that. Okay, so when a substrate binds to the enzyme, forming the enzyme substrate complex, there are two forms of binding that you want to know. One is the key and lock, another is the induced fit. That's fairly straightforward concepts. Key and lock means that this active site is exactly, it's like a circle shape, just like my substrate. So I can just fit it in right away, like a key, like a rock and a key, right? And then the induced fit is like, in my case, like let me just draw it a little bit differently. So my active site is kind of like a triangle, right? But once my substrate binds to the active site, the shape of the active site will kind of, getting adjusted, and then it will fit. Okay, all right. So when the enzyme substrate complex formed, and then it will give us back the enzyme, and then the product doesn't necessarily have to be a circle. It can be a total different compound, right? Due to the reaction that happens during this step. Okay, perfect. So now let's talk about the Vmax. Okay. So this graph that you have seen for so many times. Okay, the x-axis is the substrate concentration, and then this is my Vmax. Okay, so Vmax and V initial, VO, they can be intuitively understood, right? Vmax is just when the enzyme works, the work of enzyme reaches the maximum velocity. Okay, so I just keep adding my concentration of my substrate, like here. Okay, I keep adding, and okay, to a point, no matter how many more I add, it doesn't change the Vmax. Why? Because I have certain amount of enzyme. No matter how many more substrates you have in the system, you can't get attached to another enzyme. So when an enzyme is saturated, then we reach the Vmax. Okay, and then there's a concept called a half Vmax. Okay, so it's like let's say Vmax here it's 10, right? So half Vmax is 5 here. Okay. Is 5 here. So when you reach the half Vmax, and then the substrate that that it requires to reach the half Vmax is my Km. Okay, so Km once again is basically the substrate concentration at half Vmax. Okay, so it's really a what should I say, inherent characteristic of this enzyme. Okay, so what does that mean? So when you think of Km, I want you to instantly thinking about affinity. Affinity of what? Affinity of the enzyme and its substrate, like how tightly they can bind to each other, the thickness between the enzyme and the substrate, okay, are called affinity. Okay, so the Km is always inversely related to affinity. So when an enzyme has a pretty low Km, okay, let's say low Km, I need a pretty low concentration of the substrate at half Vmax. That means my affinity is pretty high. So I don't need a ton of substrates, right? I have a low Km. On the inversely, if I have the Km of that enzyme is really high, that means my affinity, the affinity between the enzyme and its substrate is fairly low, right? Very low affinity. That means you need a ton of substrate concentration to reach half Vmax, right? Okay, perfect. So that's Km. Okay, that's Km. Okay, now let's talk about kcat. Okay, so kcat is really, let's use a different color. Okay, so kcat is really, cat means catalyze, right? So kcat here, the lowercase k, okay, lowercase k is the it means rate, whereas the uppercase k, km here, it's like a characteristic, like remember ka, kb, those, you know, those pka, pkd, that k, also like the uppercase km is the characteristic of an enzyme, an acid, or a base. All right, and then whereas the lowercase k, it's a rate. Okay, so kcat is basically, okay, the rate when after the complex is formed, like how fast does it turn into a product. Okay, all right, so here we can also have a like a K association, right? The rate, you might see that on MCAT, right? Some data give you, okay, how the K association of this enzyme involved reaction, then you know, okay, it's how fast the substrate and the enzyme associate together. Or this can be K dissociation. Okay, perfect. All right, so now we know kcat. Okay, now we know kcat. Okay, so now we can talk about catalytic efficiency. Okay, so let me just use a different color. Sorry, I know that I have a million colors on my study guide. Okay, so catalytic efficiency is kcat over km. Okay, there you go. So catalytic efficiency is always directly related to kcat and inversely related to km. Okay, so if I put a CE here, means catalytic efficiency, you will see, okay, oh, here km is low, so CE would be high, right? Because they're inversely related. And then kcat would be pretty high, right? kcat would be pretty high as well, because kcat is always directly related to catalytic efficiency. Okay, so in this case, if km is high, okay, my catalytic efficiency is inversely related, so it's low, and then my kcat, sorry, I always miss the C. So my kcat, once again, let me just write again. It should be lowercase, okay? kcat. Okay, so kcat is always the same as the catalytic efficiency. They're directly related. Okay, so here you go. That's the relations between the variables here. Okay, and then besides these, on MCAT, you may also see something like something that you don't see on your biochemistry class, which is Vmax equals to kcat times number of enzymes. Oh, excuse my handwriting. So you've never seen, I've never seen this on my biochemistry classes, but this showed up on MCAT, okay? So as the number of your enzyme numbers decrease, let's say in vitro, right? The Vmax will increase as well, okay? So to reflect it on the graph, this is Vmax, and then this is S, and then this is a certain amount of enzyme, and then when I increase the number of enzyme, my Vmax would increase. But as you can see, my Km at half Vmax doesn't necessarily increase. Okay, I hope that's helpful.